So it turns out there were some very powerful tools that were developed that have allowed us to do these drug screening programs. One of this was a tool developed by Scott Dindot when he was in Art Baudet's lab. He made a mouse, and once again, this is just genetic trickery that we can do through genetic engineering in mice. This isn't something that would happen in humans. He engineered a mouse so that every time a gene copy of UB3A is active, the protein it produces is fluorescent. Okay, so the protein will glow, and the neurons expressing that protein will glow. So he attached what's called a yellow fluorescent protein, which is YFP, and we'll either image it as yellow or green in the following slides. But when this fluorescent reporter is bred onto the mice, so it's inherited from the mom, every neuron that expresses UB3A, we know expresses UB3A because they're glowing, right? They're this yellow color or a green color. Next slide. No, back. If you put the fluorescent reporter on the paternal copy, you can see that there's no fluorescence at all. Okay? There's absolutely no fluorescence. So this tells us that the protein that's being made is being made off of mom's copy and not the dad's copy of the gene. Okay? So this fluorescent reporter trick can allow us to distinguish between an active or an inactive UB3A gene. Okay? Next slide. So then the drug discovery project becomes relatively straightforward at this point, in that we took mice that had the fluorescent reporter on the paternal copy. We then extracted neurons from these mice and then plated them out in a 384 well format. So this is just a dish where neurons are grown in 384 different wells. Okay? Now, next slide. The idea then is to add drugs, and we add them in quadruplicate. And the reason we do it in quadruplicate is it increases our chances of not having a false positive. And we're looking for a set of 4 wells where all the neurons are glowing, okay? Meaning that we've activated the paternal copy of the gene. Okay? So the only way we can get fluorescence, in theory, from this is that either the drug itself has fluorescence, which actually happens sometimes, so we got a lot of false positives just from drugs having fluorescence, or if we turned on the paternal copy, because that's the only place that we have the fluorescent reporter. Is that clear? Okay, great. Next slide. Now, I wanted you to appreciate that these studies aren't easily done. They're really, really challenging. So one, we have to, every time we screen neurons, we unfortunately have to kill a mouse because we cannot do the study on neurons that divide. Neurons are not dividing cell types, and they're the only ones that have this gene silencing in them. Okay? The other, go back. The other is that you really need to have expertise in drug discovery, and you also need the machinery to do it. So a lot of the machinery that we used is typically found in a pharmaceutical company. And once again, we are fortunate enough to have Brian Roth at our university with his expertise. And if I don't, well, actually, I'm going to take over control. If that's okay. I think I prefer. Hope perhaps this video will work. Perhaps it won't. I wanted you to see if it works. I wanted you to see a video showing some of the machinery, because it's million-dollar machinery, and very few groups have access to this type of machinery. Let's see if it works. So you can see it's called fluid handling robotics. So these robots are actually moving around all the drugs. They're transferring the drugs into the wells that have the different neurons in them. And obviously, this can move much faster than an army of technicians. Right? So it's really, really important to have this type of technology to move forward these high throughput or relatively high throughput screens. So I think this gives you a flavor of the robotics, and you can see kind of how they act and what they look like, which I think is kind of neat. All right? you got to listen to the soundtrack too. Now, this is a painful slide for me to show because it makes a very, very long story that involved a lot of money, a lot of time, and extreme amount of effort from a lot of people into one slide. So this is looking at the changes in fluorescence that were observed, which is on the y-axis, across the over 2,300 compounds we tested originally. Out of all these compounds, only one compound, irinotecan, showed an increase in fluorescence that was significant. Okay? So we tested 2,306 compounds. We've now tested more. But from this original screen, one compound worked. Okay? Now, this compound, it turns out it's already an FDA-approved compound. It's what's known as a topoisomerase inhibitor. And these are compounds that are currently used to treat cancer. In fact, it's probably the most common cancer treatment is to use some type of topoisomerase inhibitor. Now, it's important to realize that as you probably all have had the misfortune of watching someone be treated with a chemotherapeutic, okay, you know that chemotherapeutics are not without side effects. And that's a very important thing to keep in mind, that we know what's an advantage. We know how well these drugs are taken by humans. We know what doses they can tolerate, but we also know that they have side effects. Right? So it's probably not a type of compound you would want to treat someone chronically with if they were at the same high concentrations that are used for a chemotherapy. Right?
So, just to really clarify my very simplistic understanding, this compound, irinotecan,
yes,
is causing the paternal gene to become active and to express the UB3A.
Exactly.
And it's used in chemotherapy as a chemotherapeutic in cancers currently.
Correct. Okay. Yeah. So we see that it turns on the paternal copy of UB3A, and it's already approved by the FDA for use to treat cancer. Okay? So for several different types of cancers. Okay? Now, this is just showing you that an advantage of a drug discovery approach is that once you have one compound that works, it gives you an inroads to see whether other similar compounds might also work. So you can look at molecules that have similar structures or that hit the same molecular target. Okay? So while our first study found irinotecan, once we had that knowledge, we could test a whole host of other topoisomerase inhibitors or similar molecules that look like topoisomerase inhibitors to a medicinal chemist. So this is another compound called topotecan. And this is looking at UB3A expression off the paternal copy, normally with just a vehicle, which is just the saline control, basically. We don't see any fluorescence. We know there are lots of neurons here because this is a nuclear stain to the same panel. We can see all the neurons that are there. Now, we treat with topotecan for a few days. All the neurons are expressing UB3A from the paternal copy of the gene. Okay? Now, I don't want to go into the details of this, but this is just basically a dose-response curve. At what dose can these drugs be used? And what it's showing you is that topotecan is even more effective than the compound we found originally, than irinotecan. So it works at a low nanomolar level. And this would make it so this could be a clinical candidate to be brought forward in Angelman syndrome. But of course, it has some limitations that I'm going to spend some time talking about in a little bit. Now, okay, it's great and wonderful to show that a compound can work in cultured neurons. But of course, what you really want to know is, can it work in the mouse? Can it work in a living animal? So to address this, we gave intrathecal injections of topotecan to see what the effects were of these drugs on paternal UB3A expression. Once again, we're using the same fluorescent reporter mouse to determine this. Now, next slide. Why would we inject intrathecally? Well, it turns out, and I should tell you, intrathecal is just going into the spinal cord, right? So for women who had an epidural during childbirth have already experienced that route of drug delivery. So topotecan is already used intrathecally in patients to treat brain cancers. So it could be a very ideal route for drug delivery in individuals with Angelman syndrome. So we already know the extent to which these compounds are tolerated by individuals who are being treated with cancer. So this gives us some insight into how well it's tolerated in humans. And interestingly, it's particularly well tolerated in pediatric patients over adults who are being treated for cancers. Next. So the first thing is just to show that UB3A is imprinted in spinal cord, and the imprinting just means that it's only expressed from the maternal side as it is in brain. This is just a slide showing you the lack of paternal expression of UB3A in a small section of spinal cord. Once again, we know that there are lots of neurons there from this blue stain. Now, that's just an animal that was untreated with drug. Now, go forward one. This will show you that we've activated UB3A in the mouse spinal cord from, this is a 2-week treatment that we gave to the mouse from intrathecal injections once every day. One of the limitations that I'll keep on coming back to is we're not getting a level of activation that we really want in the spinal cord and brain. If you advance the slide, this is a mouse that hasn't been treated with drug, but as you can see from the schematic above, this is with the fluorescent reporter on the maternal copy. So this is what we want to achieve. This is the UBE expression you could expect to get from the maternal copy of the gene. This is the more limited number of neurons we're getting it turned on in the paternal activation of the gene with drug. Okay? So this is a limitation that I'll keep coming back to. Okay.
Can I ask the mice that you're injecting there, they're normal mice, they're not actually Angelman mice?
These are not Angelman syndrome mice. They're still not normal mice in the sense that they have this fluorescent reporter genetically engineered into them, right? So they are genetically engineered mice. We've done these same types of experiments in Angelman syndrome mice. We actually, we now have a stain for UB3A working really well, so we no longer have to use the fluorescent reporter mice, and it also works in Angelman syndrome mice. So it's not unique to these fluorescent reporter mice. Did that answer your question? Yeah. Any other questions? Yeah.
How much do you have to give, you saying about the dosage of the drug?
Yeah.
Do you have to give the mice a lot to get that effect, or, you know, a
Yeah.
reasonable amount?
So the more we give, the more we unsilence. When we're getting a lot more unsilencing, we're also beginning to develop some toxicities in the mice.
Right.
Okay? So we've scaled that back for some of the studies I'm going to show you, but we have some other approaches to try to overcome this problem of getting into the brain. Really, it's a brain penetrance issue. So while this drug can get into the brain, it doesn't get in as well as we'd like it to. Yeah. Yes?
How long does the unsilencing last for?
You've asked my next slides. So, so is UB3 unsilencing transient? So if it's transient, it means you treat with drug, you turn on the gene, you take away the drug, and you lose UB3 expression again. To address this, the experiment we did was to inject intrathecally into the mouse, and IT is just for an intrathecal injection. We go back. So we injected for 2 weeks, so once a day for 2 weeks. Then we looked after, either immediately after a drug treatment, 4 weeks, 12 weeks, or 52 weeks later. So basically a year after we had stopped giving the last treatment of the drug. Okay? Now, I should also point out that we scaled back the concentration of the drug we're giving for these experiments for 2 reasons. One, this allowed us to avoid any potential toxicities in the mice. And 2, it allowed us to do a very an automated quantification that was less intensive than if we're can't individually image the neurons. So a computer can do it rather than us doing it by hand. So this is using low concentrations of the drug. So you won't see as strong as activation as you saw on the previous slide. Next slide. So it might be a little difficult to see this, and I'll show a blow-up. I don't know if we can, well, hopefully you can see it. If not, I'll show a blow-up on the next slide. I think, maybe. Oh, well. Hopefully you can see it. this is an untreated animal. You can see that there's really little, if any, paternal UB3A expression. All right? And here are mice that we treated with topotecan. So you can see a number of neurons expressing paternal UB3A immediately after injection. But what's particularly remarkable is essentially a year later, we still have unsilencing. Okay? So even though these mice have not been given the drug for a year, the gene is still turned on.
And the drug just administered once?
The drug was administered for 2 weeks straight early on, so those first 2 weeks, and once a day for 10 out of 14 days.
Can I just clarify as well? It doesn't turn, it's not all or nothing, it just turns on some of the cells, but not everything.
It is just turning on UB3 expression in some of the cells. So in the cells where it's turned on, it seems to be strongly activated. In the cells where we don't see it turned on, we really don't see anything for the most part. We haven't quantified this exhaustively, but just from looking at it qualitatively, that's what it appears to be. So if you go to the next slide, that's just a quantification showing that it stays on for a year. Next slide. This is a blow up, just because I think it's really remarkable that you can keep an animal off a drug for a year and still have unsilencing of the gene. So this is without drug. This is a year after the last drug treatment. And you can see that we still have paternal expression of UB3A that's on. So this is remarkable because it suggests that there's a potential, at least, for a one-time treatment opportunity to lead for a lasting, perhaps even lifelong effect. Next slide. Now, once again, you could say, Ben, this is great. You've shown that you can turn on paternal UB3A in a mouse. But we don't have mice for kids. We have humans for kids. So what about that? Can this work in human neurons? So we all know that while there are many genetic similarities between mice and humans, there are also some pretty important differences that we have to appreciate. In fact, one of the number one reasons that drug trials fail is that what is found out in a mouse doesn't translate well to a human. Okay? So this is not something that I think is in any way ready to try in humans, nor would I advocate for being tried in humans. The only opportunity that I think it should be tried in an individual with Angelman syndrome at this moment, if they also had the misfortune of having a cancer and were going to be treated with the topoisomerase inhibitor, well, then you have your clinical trial anyway, right? So what can we do then? Well, the next best thing is that we can work in cells that come from individuals with Angelman syndrome. Okay? So some of you may or may not be aware of this fact that it's possible to take even skin cells from a human, then to what we call de-differentiate these cells into stem cells. These stem cells are then capable of, through kind of molecular, thank you so much, through molecular trickery, you can force these stem cells to differentiate down a path to become neurons. Okay? So this, once again, is not something that my lab does. So we reached out to another Angelman syndrome research who is working on patient-derived cells. So this is a really talented geneticist, a woman by the name of Stormy Chamberlain, who's at the University of Connecticut. And what she has, she's been working with cells that she's gotten from patients with Angelman syndrome. She's then transformed these into neurons to make them brain cells. And then we've tried treating these cells with our drugs and seeing if it work in these patient-derived neurons. Okay, next slide. And what we found is that it works in these human cells as well. So this is just a graph showing you in red is a level of UB3A we're trying to get to. This is a patient with no treatment of topotecan, and then with increasing concentrations of topotecan, we're getting closer and closer to the UB3A levels we'd like to get. Okay? So it works in humans just as it does in mice. I will give a caveat that it doesn't appear to work as well and we think we might understand why it's not working quite as well. So it works, but we seem to need a little bit higher concentration and for a little bit longer. Okay?
And you said earlier about the basic research about the age. Has it been done with the cells of different age?
Yeah. So these cells, I don't know when they're getting the fibroblasts from, but then the neurons, when you in the process of making them stem cells and then differentiating them back into neurons, they re-go through a developmental progression in a dish. Right? So they basically start at a very early developmental stage and then go through more or less the same stages of development all over again. So these would still, even though these are probably, you'd have to ask Stormy, I think these are like 14-week-old cultured neurons. They've been cultured for 14 weeks or so. What that corresponds to in a human development would be hard for me to say. I would venture a guess that they're still relatively immature. So we're excited by the fact that these drugs can work in patient-derived neurons in a manner that's at least similar to what we see in our mouse neurons. Next slide. So I wanted you to understand some of our current limitations and directions. One of the big limitations that I didn't show you data for is that, at least in too much detail, is that the drug topotecan, which is our currently one of our best compounds, it does not unsilence UB3A throughout the brain. So even if we infuse the drug directly into the brain, it spreads out near the point of infusion and has unsilencing there, but it's not getting out everywhere we want. Okay? Or if we inject it in the spinal cord, we can see some evidence for unsilencing in the brain, but it's just not widespread, so it's more scattered neurons. We really think that this would hamper our ability to see a behavioral improvement at this point. Okay? So what we need to do is we need to identify additional topoisomerase inhibitor-like compounds that get into the central nervous system, that get into the brain even better than the current compounds we have. All right? Now, we also need to determine off-target effects that may limit the chronic use of these topoisomerase inhibitors. So we know how well these compounds are tolerated given short use, and even actually some semi-chronic applications that have been used to treat certain types of cancer, that these have been given across even 2-year schedules. But we don't know what it would be like to have long-term use. We don't even know yet if we need long-term use or not. I can tell you that we've been looking very closely at what other genes are affected by these drugs. And surprisingly, this hasn't been done very carefully by the people studying these drugs for cancer. So they just know that it works for cancer. They know that it kills these dividing malignant tumors. But they haven't very carefully looked at what other genes they're affecting. Okay? We know that how well they're tolerated, but we don't know what other genes are affected. So we've been doing a very exhaustive look at what other genes we might be affecting and whether this might have any kind of untold side effects that we are not anticipating currently.
Is it correct then to say that there's some concern about autism?
What's that?
Is there some concern about autism?
Yeah, there's certainly a concern about autism. So one concern about autism would be if you overexpress UB3A, for example, then then we know that overexpression of UB3A has been strongly linked to autism, right? So what we're seeing so far is that we're not getting overexpression of UB3A in our Angelman syndrome mouse model. It seems to go back up to neurotypical levels. But this could come into play, for example, for individuals with uniparental disomy. Then you might have a potential to turn on 2 copies, for example, right? So then, of course, then there's a higher level of ethics you would get into as well. Well, what would you change an individual Angelman syndrome to autism? I mean, that's a huge ethical issue that I'm personally not prepared to answer, and I'm not sure if anyone in this audience is, but these are the types of concerns that could come into play, right, depending on the type of Angelman syndrome an individual is carrying, right?
Are there any mouse models for UPD being looked at at all? Or
I'm sorry?
are there any mouse models where UPD?
Yeah, there are some mouse models of UPD. It's not something we're looking at right now. We're really focusing on the kind of the most common at the moment. Although I shouldn't say that, well, I'm not prepared to talk about some of the things we're doing on less common forms of Angelman syndrome. Now, we also need to determine how these drugs are working. So how is it that we can get unsilencing of UB3A? And once again, this is a basic research question. And, but I think it's a really important one. So whether or not topoisomerase inhibitors ever make it into clinical trials is really difficult to predict at this juncture. But I do know that if we know how the unsilencing is occurring, what mechanism by which it's occurring, we might gain some insights into other strategies to produce the unsilencing. Right? So once again, I think it's an example where we're trying for a clinical endpoint, but if we don't get that clinical endpoint by doing the correct basic science experiments, we'll gain insights that should benefit other approaches.